Welcome to section 12.2, Determining Evolutionary Relationships. Imagine standing in a natural history museum surrounded by skeletons, a bat wing, a horse leg, a whale flipper, and human arm bones. At first glance, these structures seem very different, but their underlying similarities reveal deep evolutionary connections. Today, we'll learn how scientists distinguish traits that share common ancestry from those that evolved independently, and how cladistics helps us map life's complex family tree. By the end of this section, you'll be able to compare homologous and analogous traits and explain why understanding the difference matters. You'll also explore cladistics, a method biologists use to build phylogenetic trees and uncover evolutionary relationships. These tools let us organize life in a way that reflects history, not just appearances. Homologous structures are features that may look different on the surface, but share an underlying similarity because of a common ancestor. The bones in a bat's wing, a horse's leg, a whale's flipper, and your own arm are arranged in the same pattern. That's a clue. Bats, horses, whales, and humans inherited this bone plan from a shared evolutionary past. Homology tells us that even very different organisms are branches of the same family tree. Analogous structures might fool you. They look or function similarly, but evolved independently. For instance, the wings of insects and bats both allow flight, but insects and bats don't share a recent common ancestor with wings. These similarities arose through convergent evolution, different lineages adapting to similar environmental challenges. A streamlined body shape in both fish and whales is another example. Whales are mammals, but their body form mirrors fish because both benefit from reduced drag in water. Distinguishing between homologous and analogous traits is critical. If we mistake analogous traits for shared ancestry, we could draw the wrong evolutionary connections. Homologous traits signal a common evolutionary path, while analogous traits are the result of convergent evolution. Careful comparison, often using genetic data, helps scientists avoid these pitfalls when building phylogenetic trees. Cladistics is the method biologists use to sort organisms into clades, or monophyletic groups. Each includes a common ancestor and all of its descendants. This approach builds phylogenetic trees based on shared derived characteristics, features unique to a group that signal evolutionary relationships. Cladistics provides a consistent, evidence-based way to organize biodiversity. A clade is like a branch on life's family tree. For example, lizards, rabbits, and humans all belong to the clade Amniota because they inherited the amniotic egg from a common ancestor. A broader clade, Vertebrata, includes these animals along with fish and lampreys. Seeing life in clades helps us understand that evolution is a branching process, not a ladder of progress. Cladistics rests on three main assumptions. First, all life descends from a common ancestor. Second, speciation occurs through splitting lineages. And third, traits change over time. These assumptions may seem obvious, but they're powerful. They guide scientists in constructing hypotheses about how traits evolved and lineages diversified. Descent from a common ancestor means scientists can group organisms into clades based on shared traits. For example, lizards, rabbits, and humans all share the innovation of the amniotic egg, so we can confidently place them together in the clade Amniota. Speciation by lineage splitting allows scientists to model life's history as a branching tree rather than a linear ladder. When two populations become reproductively isolated, say by a new mountain range or changing mating behaviors, scientists infer a branch point, as seen in the divergence of African cichlids or Darwin's finches. Trait changes over time let scientists compare ancestral and derived characteristics to decide which organisms belong together. For instance, the appearance of feathers in theropod dinosaurs marks a shared derived trait linking birds to their dinosaur ancestors. Together, these assumptions help biologists build phylogenetic trees that are not just diagrams, but testable hypotheses about how traits evolved and lineages diversified. If new evidence, such as genetic data, conflicts with the proposed tree, scientists can revisit the assumptions to refine their understanding of evolutionary relationships. To build a phylogenetic tree, scientists compare shared derived characters, new traits unique to a group, with ancestral characters inherited from a distant ancestor. For example, organisms with an amniotic egg form a clade because that trait evolved once in their common ancestor. A principle called maximum parsimony favors the simplest explanation with the fewest evolutionary changes. In science, the simplest path is often the most likely. Imagine two possible evolutionary trees. One requires 6 trait changes, and the other requires 7, including 2 independent evolutions of a bony skeleton. Both fit the data, but parsimony says the first tree is better. It's simpler and avoids unnecessary complexity. This principle helps scientists choose between competing hypotheses when the evidence isn't clear-cut. Cladistics isn't just academic. It has real-world applications. Tracking COVID-19 evolution in North America between the years 2020 and 2024 used cladistic methods to map viral lineages and predict changes. Cladistics informs vaccine development, guides conservation decisions by identifying distinct evolutionary lineages, and even helps trace disease origins to control outbreaks. Homologous traits point to shared ancestry, while analogous traits reflect convergent evolution. Cladistics sorts life into clades, revealing evolutionary relationships. Maximum parsimony keeps our hypotheses simple yet powerful. Together, these tools allow scientists to reconstruct life's history and apply that knowledge, from conserving species to fighting diseases.